Okay, so well, let's get started. One of the first things that we always do is because this is being recorded. Is this being recorded, Mia?
Yes, it is.
All
It
right,
sure is.
so because it's being recorded, and will be available on YouTube. What we always like to do at the start of every module is to note that this is being made available under the Creative Commons share alike license. um, which means that you can use it for your own purposes so long as the original creator is credited. Okay, so moving on to our module today, we're going to be talking about genome assembly and annotation. Um, and this is something that I have been involved in for a good part of my career, more on the annotation part than on the assembly part. So I'm happy to run you through it and also be kind of steering you in the lab towards some of the work that I've been doing with some of my collaborators on working with annotating genomes. So the learning objectives for this module is to understand the process of assembling a genome de novo. Um, and so Jared has already covered the reference mapping. That's one type. Some people consider that to be a type of an assembly, reference-based assembly. We're going to study, we're going to take a look at some genomes without a reference. Okay, and so we're going to take a look at some of the different strategies for doing those assemblies for prokaryotic genomes. So prokaryote, I think for people taking a microbial genomics course, it's probably not necessary to explain it, but for just to make sure that everybody's on the same page, a prokaryote is a free living organism that lacks a membrane enclosed nucleus or any other membrane bound subcellular organelles. So that includes bacteria and it also includes the archaea. And we're going to be focusing on bacterial genome assembly and annotation, but the concepts and the techniques that I'm describing here apply also to archaea, so prokaryotes in general. Okay. So we're gonna follow look at some strategies for assembling them and then assessing the quality of the assembly. Then we'll move on to annotating the genome and and then also a little bit of using those annotations to assist with the quality assessment, but more actually for just understanding and aligning understanding and interacting with the genome that you generate. And that's what we're going to follow up with the lab is we'll be looking at some of the tools that are available for assembling and annotating prokaryotic genomes. Um, but Before we get into the nitty-gritty, I wanted to just go quickly over genomes, some of the characteristics of genomes that will be important for some of the stuff that we're going to be discussing in the lecture and the lab. So again, most of this will just be review, I believe, for the students, especially given some of the research that I can see that you guys are involved in that you reported on today. But just to make sure that everybody's on the same page, just wanted to let you know to say that to make clear that genomes are essentially just the entirety of an organism's hereditary information. People think it's like, oh, it's the DNA, right? But it doesn't need to be DNA because some organisms encode their genomes in RNA. And so that's inherited, and it's not just DNA, right? So for example, HIV has an RNA genome. So we have here microbes like bacteria and you can microbial eukaryotes, and we have DNA-based genomes, double-stranded DNA. Um, and the But regardless of the medium that's used to encode that hereditary information, the genome itself basically includes these genes, and the genes can be of, well, two types. There's these coding genes. Those are ones that they're called CDSs. They code for proteins, and then there are other coding genes for RNA like tRNA and ribosomal RNA. And then we get our non-coding sequences. So these are also typically RNA, and these would be like non-coding RNA, small RNA, silent RNA, those types of things. Okay, well, let's take a look at some of the characteristics of the bacterial genome. So the bacterial genome is highly compact and efficient, and is optimized for bacterial lifestyles for rapid growth, adaptability, and efficient gene regulation. So most bacteria have a single circular DNA chromosome, like shown here at the bottom for Yersinia pestis, one single chromosome. But some have additional chromosomes. So like Vibrio cholera here, Vibrio cholera has two chromosomes. Um, this is typically it's kind of rare, um, but it does happen on occasion when you have more than one chromosome. It's typically what's happening is that a plasmid has become essential, and has integrated its replication cycle with the main chromosome's replication cycle. And so essentially, basically, it's kind of like sort of like a super plasmid. It's evolved and is now under control, replication control from the main chromosome. So formally becomes a second chromosome, but it was back in its day, it was just sort of like a super important plasmid. And that's typically what you see for these types of multiple chromosome genomes. You can also get uh, polyploid genomes. So most bacteria are haploid, which means that they only have the single chromosome, and the genes are available in one copy, are present in one copy, but um, there are some species like the like Diplococcus and Dinococcus that can have more than one copy of their genome under certain conditions. But we're just going to look at the haploid. Today, we're just going to be looking at haploid, um, single chromosomes. Yeah, I think that's just about all we're going to be looking at today in the lab. All right. So the bacterial genome size can vary quite a bit, and also it is tied to their lifestyle. So the smallest genomes on the order of about 1 meg or under a meg, about half a meg for the smallest known free living organism. That's the Mycoplasma genitalium. Oh, actually, it's not quite free living because it's an obligate parasite, but it only has a half a meg genome. And so the idea here is that some genomes, if they live in these nutrient-rich, stable environments, they may have metabolic pathways and other genes that are not really necessary for them. They may be essential, but it's not necessary for them to generate themselves because it's being provided from the environment. So like a host cell is a good example of that. And so they will start to accumulate errors in their genes that their genes are no longer needed, so they will become inactivated. These are called pseudogenes. Over time, they can become deleted because there's just not energetically favorable or evolutionary favorable to maintain inactivated genes over time. And so that will result in a shedding of the genome structure over time and a reduction in the size of the gene. Horizontal gene transfer, in contrast, which is the acquisition, not through vertical transmission, but through some horizontal transmission, um, is a method for acquiring very quickly large amounts of genomic material. And so the organisms that are kind of like live in free living environments and complex environments like the soil, they tend to engage in a lot of horizontal gene transfer. And so they have variable genomes, but they can grow quite a bit. And they're typically between about 5 and 10 megabases. E. coli is a good example of a free living organism that is, about 5 megabases. That's about an average genome size. And then in some cases, you can get really large genomes that have acquired like really like entire uh, not just metabolic pathways, but like super pathways and stuff like that. So they can be quite large, and so that. So that basically can influence the approach to the way that you assemble and the way that you annotate and analyze your genome. Okay. So all bacterial genomes have an origin of chromosomal replication. Actually, eukaryotic genomes do too, but they work differently. So the OREC is this origin of chromosomal replication in most bacteria, and this is a very specific sequence that's um, its purpose is to provide the site for the initiation of replication. So it's essential, and um, it's basically there to make sure that the replication cycle is carried out accurately. And it contains these important features, these AT-rich motifs. Talk about on that slide. But eight's later. But AT is that's the AT bonds are thermally unstable bonds, and so this makes this region unstable. It makes it easy to unwind to start the replication procedure. And it also has these DNA boxes. So DNA is the gene that is responsible for the initiation of replication, and it's its own regulator. So what will happen is it will if there's lots of it around, it will basically suppress itself. It's a repressor, it's a negative repressor. But when you duplicate the genome, these DNA binding boxes will duplicate, and they essentially what they'll do is they'll pull the DNA repressor away from, or the DNA, a transcription factor away from its repressor, and so that will basically help to get replication going. So that's the reason why you have this structure here, and that is essentially the ORC, which is up here. And so then you get your replication, which proceeds bidirectionally. So you have our leading strand and here, and then you have your lagging strands. We'll talk about that in a second. And then it proceeds until you get to the terminus of chromosomal replication. It's called the terci, and there you have your two daughter cells. Okay. Oh, and I also wanted to mention that the reason why I'm spending time on this is because a lot of genomes, bacterial genomes, will assign the first base in their numbering scheme. So they're circular, but you got to pick one place to be the position number one, and they'll typically use in some, not in all cases, but sometimes they'll use the OREC. And the DNA gene is almost always located adjacent to the OREC because their proximity is a important part of the regulation. So if you find a DNA, if you're looking for the OREC, these are not really strong motifs, but the DNA gene beside it is, so it will help you to find the REC. Okay, so another aspect of well, genomes in general, but is an important characteristic for bacterial genomes is the GC content, and so this is the percentage of the G's and C's relative to the total number of bases, A, T, G, and C. So there's the formula. Most people are familiar with this formula, and we know that this is important because G's and C's have the 3 hydrogen bonds, so they're thermally stable. These are called stretches of G's and C's are called GC clamps. And then the AT only has the 2, so these are less thermally stable. Just like I mentioned, we have these AT boxes that are used to help to unwind DNA. But the presence of differential GC content, like we see here, we're plotting the GC content, and you can see the GC content is kind of like average about 50% here, but then it kind of goes down over in this section here and kind of goes back up again. And this coincides with the insertion of a phage element. So a phage is a protein that attacks bacteria, can insert itself into the genome, and that's what has happened here. And because it's foreign DNA that's inserted itself into there, it has basically a GC content that is not has evolved differently from the source organism than the one that it's being inserted into. And so the GC content, which is a kind of like a marker, and is characteristic for large bacterial groups like the phyla, they can receive um, uh, genomic content from other organisms that may be from different phyla, and these will have different GC content. So that basically that GC content can be used as a marker for helping to identify horizontal gene transfer, mobile elements. Okay, so GC skew. It's kind of similar to GC content, but it's a little bit more nuanced. So where GC content is measuring the GC composition as a function of over both strands, and the total gross composition of your genome. GC skew is measuring the imbalance between the guanine and the cytosine, the G and C, on one strand versus the other. And so this can arise because of the different mechanisms used to replicate the bacterial genome on the leading strand versus the lagging strand. So the leading strand here you can see is a continuous synthesis. So you are unwinding, and then the polymerase is following the helicase, and it is continuously adding the second strand. On the lagging strand, it is discontinuous. So you have what are called these Okazaki fragments that are being um, uh, placed onto the lagging strand, and then they have to be primed. So they have a primase, and then they have to be ligated with a ligase. So in addition to the polymerase, there's the additional enzymes, and those can introduce errors. And one type of error is called a cytosine deamination error, and that can result in a C to T transition, and that is a where you can get this imbalance. Also, the lagging strand spends more time in a single-stranded state than the leading strand, and the single-stranded DNA is more prone to chemical attack. Than double-stranded. And so that when it's in a single-stranded mode, that means that the C is more sensitive to enzymatic degradation to the cytosine deamination. Anyways, the reason why I'm going into all of this is because well, GC skew is also a measure for identifying, or it's a signal for identifying possible horizontal gene transfer, but it can also identify the origin of chromosomal replication, because you'll see a switch here from on the leading strand versus the lagging strand. So the GC skew will go from one polarity to the other very abruptly at the origin of chromosomal replication, and then it will again do the same thing at the terminus of the chromosomal replication. So you can look for these to try to help to identify the origin and that can help you to orient your genome. Also, if you get switches in versions where parts of the genome just flip themselves over, then that will flip the skew, and so you can identify inversions by looking for these abrupt changes in the skew. All right. You know, I want to cover a little bit about the bacterial gene itself. So, the bacteria. Okay, I think maybe the best way to understand this slide is to recall that a bacterial gene encodes a protein through two steps, right? One is transcription, where the polymerase transcribes part of this genetic region into a transcript that you see here. And then that's our messenger RNA or mRNA. And then the ribosomal machinery is going to translate that transcript into a coding sequence here. So the first step is generate a transcript. The second step is generate a coding sequence. And the bacterial gene contains the instructions for doing both. So we have a promoter region over here, and here's a blow up of the promoter region. So the promoter region contains these two motifs called a -35 box and a -10 box, or a Prebno box. And these help to position the polymerase right at the transcription start site here, which is just downstream of these positional boxes. And so they're there to ensure that the first nucleotide is correctly transcribed into the RNA. Then you will continue past this 5' untranslated region here. Helps for stability and the amount of expression that the mRNA is going to get, but also contains what's called a ribosomal binding sequence or a Shine-Dalgarno sequence, and we'll talk about that in just a sec. And the transcript also includes the coding sequence, and it goes past the stop codon at the end of the coding sequence, and then and will continue into what's called the 3' untranslated region here until it encounters a transcriptional terminator. So this provides signals to end transcription, and so the polymerase will fall off at this point, and that's what generates the transcript, the mRNA transcript. Then the ribosome will align itself to the start codon of the coding sequence on the mRNA transcript by binding to that's ribosomal binding site of the Shindell-Garnett sequence. That will initiate the translation of the transcript into amino acids, right? The protein, the polyprotein, and that proceeds through this coding sequence until it gets to the stop codon. And that is what is ultimately, well, there's some possible post-translational modifications, but that's essentially the process basically of generating a protein from a bacterial gene. And those are all the components that are required to do so. Okay, so that covers our little whirlwind tour of the bacterial genome. Now let's move on to assembly. So Jared has already covered reference assembly. I'm not going to talk about that. But again, that's where you have a reference to help to place the reads on and into a pile up, and that gives you the evidence that you need to be able to look at the differences between your reference genome and your newly sequenced genome, and to extract out those differences, which are typically in the form of single nucleotide variants or indels, or basically single evolutionary events. So you can get like an introduction, maybe a small, like, the three nucleotides, like a codon, as a single evolutionary event. So when you don't have a reference genome, or you have a genome that doesn't that is not really close to anything that is already previously sequenced, then you may wish to perform de novo assembly. So you don't have a reference, and you're just going to use the information that's contained inside the reads to try and generate your assembly that way. Let's see how that's done. So there's these 7 different steps. One is pre-processing, and Jared already covered this. This is the removal of contaminants and low quality reads, your adapters, etc., and just getting your reads to the point where they're ready to be assembled. Then there's an overlap detection step where we're going to look at every read and we're going to compare it to every other read, and we're going to identify the overlaps. And so because there you have internal repeats, you can start to, you may have uh, read may have more than a connection to more than one other read, right? And we're going to take a look at that in the graph construction stage. So once you have those overlaps, you use them to build this connectivity graph. Once you have that, and so I'll show you on the subsequent slides. So it's missing here. Then you traverse that graph to try to find the path that represents the contiguous read, the best possible overlap of, because essentially what's called the shortest common superstring of all of those overlapped reads, and that's your draft genome. And that's where most people stop, and that's where we get here. So um, we have these 3 contigs that we obtained from our original genome and one internal repeat, but we don't, because if the reads are smaller than the internal repeat, then we don't have it can't capture the flanking diversity on either side of an internal repeat. So it can't make connections between the contigs that are being generated outside of the repeat region. And because of that is the case, there's no way to connect the contigs that are intervening or interleaving these internal repeats. So in that case, the algorithm will not even make an attempt. It will basically just say, this is as far as I could get with this contig, with contig C, here's contig A, here's contig B, but I don't know what their relative order or orientation is to each other. In that case, we can use extra evidence used from sequencing to generate scaffolds. So these are, well, Jared mentioned these paired end reads where you have you're sequencing from the end of a fragment, but not all the way through. So you're going to have this internal section. There's another variant called mate pair reads where you have a long intervening stretch here. It could be maybe 4,000 base pairs. And you're just reading, maybe 100 base pairs on the ends, but because this distance between them is known, they may span an internal repeat and can capture enough unique genetic content in the contigs so that it can order them either like in the direct ordering or flip them into the reverse complement and connect the and so it can order and orient them together into this kind of like a it's not a completely finished genome, but it's a basically it's an ordered and oriented scaffolded genome. Then the final part is to close the gaps, gap closing, and you do that by generating small reads over these gapped regions and sequencing them and confirming that that A does connect to an internal repeat and B connects to an internal repeat, etc. And this is to resolve that order and orientation, connect everything together, essentially ligating everything together in silico, and that's how you get your finished genome. But it's very rare that people are going to perform any steps beyond generating your draft genome, essentially just generating your context. We certainly are not going to do anything past just generating context. All right, so we mentioned there's a couple different ways to generate these graphs, connectivity graphs that we're going to have to traverse. So the two main ones are the overlap layout consensus and the de Bruin graph. We cover overlap layout consensus with the first approach that was developed. And it's pretty intuitive. The idea is that you compare all the reads against all the other reads and you identify the overlaps between them. And then you can use that to construct a graph. So in this toy example that I have over here, we have our internal repeats, which are longer. So it looks like 1, 2, 3, 4, 5, 6, 7, 8, 9, like 10 nucleotides in length, but our reads are only 1, 2, 3, 4, 5. So we know that our reads can't span the internal repeat, and so that they're not going to be able to do the resolution. But if we do compare like all the overlaps, we can start to connect them up. And in this toy example, you can see that they connect. There's only one connection between the reads until we start to get to this repeat region here, and things start to get more complicated. So in this one, we can continue for the first repeat, we can continue walking through the repeat, but when we get to this read here, it can connect to either this one or this one, which corresponds either to here or to here, and we just don't know. So there's two branch points over here, and then this forms a loop, and this structure where we got unresolved path through through the graph is called a tangle. The last part basically is to try to untangle it to get the best representation, the best contigs that we can get out. And that's what we get here at the end. So the overlap layout consensus approach so it's pretty simple, pretty straightforward. It's computationally intensive, so it's kind of slow, but it uses a fuzzy matching approach. So when you're doing this comparison, it will tolerate differences in the overlaps between the 5' and 3' ends. And so that works really well for long reads and for uh, higher error rates in the reads because it is accommodating of higher error rates. So for things like Oxford Nanopore or PacBio, the overlap layout consensus approach is a favored approach. Okay, so de Bruin graph was this approach is a variant that was developed specifically to aid in rapidly assembling genomes from short read data with high accuracy like Illumina. So here we're going to we have our genome over here and we have a read here. So this read is, well, okay, so it's longer. It's longer than it spans this junction here, but just for the purposes of illustrating, I just want to show you how it works. So what we're going to do is decompose this read into what are called k-mers. So a k-mer is just essentially like a subsequence of your read that's a fixed size. So this is a k of 1, 2, 3, 4, 5. So I have a k-mer of k equals 5 and a step size of 1. So that means we're going to take this read and we're going to decompose it into its first subset here, this k-mer, and then we move over 1 and take another 5 nucleotides, and that's a second k-mer here, and we're gonna go all the way down here until we have decomposed the read into k-mers. Now we're going to connect these up by looking for the K-mers that have a 4 nucleotide overlap. So that means that only the 5' and the 3 on one read or one K-mer and the 3' nucleotide on the other K-mer are not going to match. The internal 4 will match. And so if we find that, then we can make a connection between that one K-mer and the next K-mer. So it's kind of like we're decomposing these, and then we're going to connect them back up. But remember, we're doing this for all reads over the entire genome and to generate all of our K-mers, and now we're just going to connect up all of our K-mers. So in this approach here, you can see we're kind of connecting things up into we have single connections all the way through here. But if the reads are smaller, we end up then the repeat length, then we end up with a similar problem with the overlap layout consensus is that there's more than one path through the graph that we generate. And we still need to be able to be able to get to resolve that. But the Bruin graphs are because they use an exact matching technique to identify the overlaps, they're extremely fast. That is an extremely fast, computationally like just blazingly fast type of comparison. Whereas overlap layout consensus uses this fuzzy matching technique, which is a lot slower and more computationally intensive. So this gives us our speed boost by breaking things down into these K-mers and doing these exact matches. But it doesn't fix the problem with these tangles that we get in the data. But because of their advantage, their speed advantage for highly accurate reads like Illumina, they're favored for doing this type of short read assemblies. Okay, so let's just take a look. Here's an E. coli genome graph, De Bruin graph that has been assembled, and you can see all the tangles in there. And you also see some of these small these called like little loops or bumps that in the De Bruin graph that come from these small little errors that, and then the algorithm knows kind of how to traverse and correct those. But you still see these like a lot of these connections, and so these are going to be all of the unresolved internal repeats that are here in our main chromosome. This is likely a plasmid, maybe a secondary plasmid, and then other unresolved repeats that are in our assembled graph here. Now, this is not the same genome, and it's just for illustration purposes. But if you have a long read that can, and the size of that read spans the majority of the internal repeats, then you will get less of a tangle, and you can, in theory, get just a single long contig if you have reads that are longer than all of your internal repeats in your genome. You'll still get these little bubbles. And from Oxford data. You can get quite with high error rates, you get a lot of these little bubbles. But again, they can be addressed through the contig generation process. Yeah. So hybrid assembly is a process that is attempting to kind of make the best of both worlds. It combines um, the features, the best features of long reads and short reads. So essentially the idea here is that the long reads, because they can span internal repeats, they would make a kind of a great scaffold for generating a first draft of your genome. Then if you also have short read, but there's going to be a lot of errors. So here I have like an overlap. We've extended these reads into a contig, but there's a conflict in the data here. Because of an error. If you map your short read data to these contigs, then you can essentially polish it, identify, say, okay, well, that is definitely an error because it doesn't appear in my high quality read data, so I can correct it and extend and resolve the ambiguities and the errors in my genome. Okay, so we're going to move on to quality assessment. Before we do, this might be a good opportunity to take any questions, if there are any questions. I can't see the Slack channel or anything, so if there's anything in Slack, maybe the TA or someone can read it out to me or just put up. Someone could just help me out and we can we'll do it. If you have a good question, you can just ask directly over Zoom.
Absolutely. No questions in the Slack currently, but I'll keep you posted.
Okay. All right. Okay.
Can I ask
Uh,
a question, quick question?
okay, great. Yeah, go ahead.
Sorry, for the Kamer extraction, what would be the length? I mean, is there any optimum length for
Yeah.
Kamer?
Okay, well, I'm really glad that you asked that question because yes, there is the length of the femur is important if you think about why. If you think about it for a bit, the reason why should become apparent. The if you let's go to extremes. Like, let's say if we were to choose a Kamer size that was about the size of the genome of our million base pairs. Um, Well, that's too extreme. Let's just say let's say we have a 5 million base pair genome and we're going to choose a Kamer size of like maybe 1,000 base pairs. Then the errors in those the reads that are being generated, if the error rate is high enough, then every read will have an error and you will not be able to overlap any of your Kamers because they only do exact matches. So you want to get them small enough that you're going to avoid the inherent error rate of the instrument so that you're going to get a lot of matches, right? But if you make them too small, there's not enough specificity in your graph. So if you make your Kamer size of 1, then everything is either an A, a G, or T, or C, and you're just not going to be able to resolve anything. So it's a question of trying to hit that sweet spot for the sensitivity versus the specificity. And it turns out for an average bacterial genome, if you do the math, the Kamer size, so about 5 megabases, the optimal Kamer size is on the order of about 21 base pairs.
All right. 21.
Yeah.
Uh, so is there an influence of the length of reads for this Kamer selection?
Well, you
Like,
wouldn't want to make your reads lower than your Kamers, but I just don't think so because most of the back in the day they had some really small um some very very short reads. Some of the sequencers like the ABI Solid I think generated these really short reads. But in general, the Kamer size, if you're going to use a chamber size on the order of 20, it's not really going to matter. It's not going to be influenced by your read size because the read is going to be so much larger.
thank you very much.
Okay. So let's keep going. So we wanted quality assessment. So we have our context, but we want to know how good of a job did we do? So there's a so we want to assess the quality in doing so. We wanted to take into account these 3 C's. So one is called contiguity, the other is called completeness, and the other is correctness. We'll address these in turn. So contiguity is the extent to which the genome is represented in these long sequences. The original genome, remember, original genome can include the main chromosome, and plasmids. So it may be that there's the optimal assembly size is not just one contig, right? Because if you have because they're separate actual physical pieces of DNA. So you want to get them to be as long as you can, but not too long, right? So that so that the contigs are basically giving you a good estimate of your genome. So the two metrics for estimating the sort of like the length of your contigs and the quality of your contigs is the N50 and the L50. So the N50 is so it's the contig length at which half the total assembly sizes are contained in contigs or scaffolds of equal or greater length. So that's the formal description, but the way intuitively to think about it is if you take your contigs and you were to rank them by size from highest to lowest, and then you count the number of nucleotides cumulatively until you get to 50%. So if we have 100 nucleotides in our genome, we count until we get to 50 nucleotides, 25, 40, 50, right? And then we stop. And we say, okay, which contig contained that 50th percentile nucleotide? And what is the length of that contig? The length is 10. So we would say the length of the contig that contains the that contains 50% of the genome in our ranked list of genomes. That size, that contig size is the N50. The rank, the actual position that it occurs in, so it's in the third position, that's the L50. So the number of contigs needed to reach that 50% assembly size. Now you would think that if L50 is for the number of contigs, you'd call that the N50, and if the N50 is for the length of contigs, you call it the L50, but they're exactly the opposite for obscure reasons. But anyways, that's just the world that we live in. But that's N50 and L50. Let's keep going. So completeness is a measure of just how much of the genome is represented in the assembly. So, we have our expected genome size here is 100. We generate something that's, less than that size, then, we're like, well, that could be a measure of just how much, of the quality of our assembly. So we can just take a look at but to do it, you need to have an expected genome size. You may not have that, right? Or it could be that your genome is radically different just for some peculiar reason from the expected genome size. So it's not great, but it's a measure of quality of the quality assessment. And in most cases, it's going to be, pretty close. Core genes is another measure of how much the genome is represented in the assembly. So bacterial genes contain the suite of these like core housekeeping genes that exist in just a single copy. They're called single copy genes, single copy orthologs, um, and so they are distributed around the genome, and they're associated with different taxonomic groups or different lineages. So if you know the lineage, you can say, I expect to see these genes in a single copy. Take a look and see if they do. If your assembly doesn't contain them all, then that can tell you that you may have some missing content. Alternately, if you have more than one copy, that can tell you that you may have contamination. So it's not just completeness, it's also correctness. And the last thing that you can do is to just map your reads back to the assembled contigs and look to see how well do they concord. So if you find unmapped reads, I don't show them here, but in this case, if you did have content that wasn't sequenced, then you would have some, or that wasn't assembled, but was sequenced, then you would have some reads that that did not get incorporated into the assembly. And so finding that can give you that situation exists can be a measure of the quality of your assembly. Okay. And so we've covered, completeness and correctness. So correctness is just how closely the assembled sequence maps the true genome sequence. Again, the mapping the reads back to the assembly and looking for that, for alignment consistency and checking for mismatches and other errors is the way to go. Essentially just, and you can also benchmark it against a reference genome if you know if you have a reference genome that you can rely on. All right. Just a couple of quality assessment programs that are available. Quast is a very popular program. We've incorporated in this program called Proxy that we're going to use in the lab. We'll take a look at that for some of our L50 and N50 reports. Busco is a program that looks at those single copy orthologs, and so these are lineage specific single copy orthologs. If you supply your assembly and the lineage that your genome is from, and it will say, okay, well, these are the marker genes I expect to see from this lineage, this taxonomic group. It belongs to this taxonomic group. These are the markers for this taxonomic group. Let's see if they're in there, or in the expected proportions, which is essentially a single copy. And then CheckM, which is more recent, but it's also a very popular tool for microbial genome assemblies. It's also done for metagenomes and can be done for eukaryotes, but it's really designed for microbial genome assemblies, and it's gaining in popularity. It's quite a popular program. And then here's some popular assemblies. I'm not going to go through them all just to show you, but there's a combination of the OLC and the Bruin graphs. The two that I want to point out are Spades and Schizas, just because we're going to use them in our module. So Spades is it's a popular de Bruin graph based de novo assembler. Actually, it's a variant of of de Bruin grafts, but it is a very popular, fast assembler and hybrid assemblies, short read assembly, pair and meat pair. Can do metagenomic assemblies. It can do lots of stuff, and that's why people really like it. It's very accurate, but it's also compute intensive. Skiza, in contrast, is an extremely fast assembler with reasonable accuracy, but it was designed more to kind of generate rough assemblies than to generate really high quality assemblies. And we also use that to get a sort of a first sense to do the quality assessment in this program called Proxy that we're going to be looking at later. So the assemblers that we use are Spades and Skiza, but there are these additional assemblers that are popular. Fly is one of the newer popular assemblers that people are starting to use. So I'll just point that one out as well. If you're going to look at one, maybe take a look at Fly. Okay, so that covers assembly. Maybe I'll just can take a quick look at the time. What am I doing for time? So looks like what time did we start?
We started at 12:30.
Okay, well, I better hurry up. Okay, let's get back to it. How do I? Oh, it's because this thing's in the way. Okay. Well, actually, this part here isn't going to take that long. And I'll try to be brief. So we've covered the genome assembly parts and specifically on bacterial genome assembly though, but now we're going to move on to genome annotation and specifically to bacterial genome annotation. So as soon as once you generate your context, typically one of the first things you're going to want to do after that is to find out like what is inside genome annotation. Basically, is that process of identifying those functional elements within a genome. So there's the genes, regulatory sequences, and other features. So features anything that is just the noteworthy inside of a genome that has a start position and an end position. So start coordinate and end position. Something in between them that you want to describe, that is a feature. So a feature could be a gene, it could be a promoter, it could be like a larger feature, a larger element like integrative and conjugative element, other mobile elements, a phage, CRISPR, etc. Yeah. So it's but when people think of genome annotation, mostly they think about just the genes. And so the genes are basically going to be these protein encoding genes and our number on protein encoding genes. So there's two main steps to do annotation. One is to find out where they are, and then the second is to find out what they do. The first, what they are, is the structural annotation part, and that is used to find protein encoding genes and also non-protein encoding genes. Those are our ribosomal RNA and our tRNA. Once you find that ribosomal RNA and tRNA, you already know what they're doing. You don't actually have to figure out what they do. So then that's basically completes the annotation for non-protein encoding genes. But for protein encoding genes, once you find out where they are, you're not really going to know what they do. So then there's a process of functional annotation. And we're going to cover that in a little bit of detail in the upcoming slides. Let's just talk a little bit here, a little bit more, just getting everybody up to speed on the genome here, just finish it off. So remember, we have double-stranded DNA, and the genes can reside on the forward or direct strand or on the reverse or opposite strand. The genes are dense. About 75 to 85% of a bacterial genome is it's harbored by genes or genes or contains genes, and then there's only about 15 to 25% is what is called intergenic region. Those are the parts that don't contain genes, but they can be important. The genes themselves are contiguous, so in contrast with eukaryotic organisms, which have like these introns and exons that distribute that protein coding information, that they can distribute it over long regions of a genome. In a prokaryotic genome, there's a start and a stop, and it's all protein coding sequence in between, so they're contiguous. Right? Yeah. Okay. There. So. So one of the first things to do that you need to do if you want to find out where the genes are is to look for the open reading frames. And this is a very, very basic characteristic of a gene that it has to contain an open reading frame. So the open reading frame begins with the start codon. That can be like an ATG. and it ends with a stop codon, and it proceeds in nucleotide triplets called codons in frame side by side from the start codon uninterrupted until it gets to the stop codon. And so the start codon I mentioned is ATG, but it can also be, there can be some exceptions, so GTG and TTG. ATG codes for methionine, GTG codes for, I think, for leucine, and TTG for lysine, or but regardless, because of the machinery that's involved in protein synthesis, always incorporates N-formal methionine as the first base. It doesn't really matter what the code is. It's always going to be a methionine as the first base. So either it legitimately codes for methionine or it codes for something else, but it's still going to be methionine when it gets to the protein stage. Like I mentioned, there's these nucleotide triplets that encode the amino acids, and those occur side by side in frame from the start codon until you get to the stop codon. And it's also important to note that some open reading frames contain coding sequence and some do not. We have 64 codons that specify 20 amino acids, and 3 of them are stop codons, which means we have a 3 in 64 chance. If we're looking if we were to just take random nucleotide triplets and place them side by side, we would get a start codon followed by a stop codon approximately once in every 21 nucleotides, just from straight. That's without taking into account the distribution, like the natural occurring distribution of amino acids in the wild. Right? That's just completely random. So just because you have an open reading frame with a start and stop codon doesn't mean it actually encodes anything. So there's additional things that we're going to want to look for, things like a Shine-Dalgarno sequence, and also basically the content of the um, of the uh, of the open reading frame to help us identify whether it actually codes for something or doesn't. Okay, so we want to detect these ORs, but there's a problem. There's a couple of problems. One is that we want to detect all the ORs, and the ORs may occur in more than one reading frame. So because we are working with codons, and codons occur in nucleotide triplets, and that can occur on the direct frame or on the opposite frame, that means that there are 3 reading frames that we have to consider on each strand for a total of 6 reading frames that we're going to have to look at. And you would think intuitively you might go look for start codons and then follow to see if you can go in frame in nucleotide triplets till you get to the stop codon, but actually doesn't work. And that's because the start codon, which codes for methionine, can occur in the interior of a open reading frame or a gene as a legitimate coding sequence for methionine. So because methionine has a dual duty as a signal, so it codes for start and it codes for methionine, you don't know if you find an ATG whether it is the start codon or whether it's an internal methionine. So the trick there is to go backwards. So you find the stop codons, and then you will go from the 5' direction from the 3' direction in this to the 5' direction. So we're going this way from the right to the left, and we're looking for another stop codon because you can't have 2 stop codons in frame that code one gene. If there is an open reading frame that contains a gene, it must be between 2 in-frame stops. And so that is the idea. Look for in-frame stops in every reading frame, and then make these our candidate open reading frames. So here's an in-frame stop. It's from stop to stop, and there are no internal methionines, which are these half bars here. So that's an open reading frame. Maybe it's a gene. Here's a longer one that goes from stop to stop or stop to stop. And here are all the ATGs. Now, some of these are going to be internals. Some of these are going to be a start. And some might just be, you maybe get an ATG in outside of the coding region. So you don't know. Any one of these could possibly be the start codons. One of the tricks is to try and find that start codon. And then here's just another example here. So okay, so how do we know if the open reading frame actually encodes a protein sequence? So, well, one way is to look for an upstream ribosomal binding site or an RBS sequence. So remember, this is the motif that is used to position the ribosome at the start codon to begin the process of translating that mRNA transcript into a protein sequence. But this is kind of weak signal here. So you see there's a preponderance of A's and G's, so it's AGG motif sequence, but there's a lot of variation. So it's not a strong signal. So it's not the only thing that we can use to help to identify the coding sequence, but it's basically just it's one of a couple of different methods. So this is basically called like a heuristic method, and that's what modern programs these days, like Prodigal, use. Is essentially what they're doing is like looking at these rules of thumb. There are other programs that use things called like hidden Markov models to do it, and I'm going to talk about hidden Markov models in a second, but I'm not going to get into the details of actually how you discriminate an open reading frame from like the nitty-gritty of the algorithms that are used to discriminate an open reading frame from a true gene, but just to let you know that they do exist. Okay, so once you find them using a program like Prodigal or Glimmer, then you found your protein coding sequences, and there's the remaining non-coding genes, our tRNA and our ribosomal RNA that we want to find. It's just, I'm not going to go into too much. And of course, we're going to get to the end of the lecture pretty quickly here. So I'm going to kind of speed up a bit, but tRNA are small, and they have the primary sequence here. There's a lot of variability, but the secondary structure is very fixed. And so we can use that information to try and help us find them. So that's tRNA, and then the ribosomal RNA, these much longer molecules, they typically exist. They are a combination of these separate segments called like 5S and 23S and 16S and 30S and 50S, depending on whether you're eukaryotic or prokaryotic. You have these different components, but they come together to form the ribosome. They can be about 5,000 nucleotides in length as a co-transcribed operon. But that's a second type of gene that we're going to want to look for. So we want to look for these, and we want to look for these. Okay. So I mentioned that the tRNAs have a highly conserved secondary structure. So that's the bonding structure that it has to itself. Um, and whether it forms these secondary structures like these loops and stems. So, which it does. And so these are conserved, and you can look for these. So even though it may be hard to just look for patterns in the sequence because of variability in the sequence, you can look for patterns in the primary sequence, you can look for patterns in the secondary sequence, and this is very highly conserved. That can help you to identify. So the program called tRNA scan, it uses a decision tree approach to be able to find them. So it scans along your genome. And it's going to look for some of these signals. So a signal will be something like this D loop or your T-cy-C arm, right? Your anticodon signal. These are the things that it's going to look for. And if it finds them, it will add to the score. And it keeps working its way through this decision tree, looking for these different parts. And if it finds them, it will accumulate a score. At the end, if the score is high enough, then it will say, it looks like I found myself a tRNA. And that's basically it. This is a closed kind of problem. Original tRNA scan could identify them with higher accuracy. The most recent one, which is tRNA scan SE, has a accuracy of about 99.5. It was up so one error in 4,000 average genomes. So it's very accurate. Ribosomal RNA prediction is less accurate. It uses a different approach, and essentially what the idea here is that we're going to use it's kind of like an AI approach, a machine learning approach called the hidden Markov model, to try and identify these longer stretches, these RNA operons. So I don't really have time to get into detail about hidden Markov models, but the way to think about it intuitively is that it is a probabilistic model of a collection of sequences that are related through homology. So they're evolutionarily related by a common ancestor. And they're fed into you train a hidden Markov model by supplying it with a multiple alignment. So column by column, it's going to go through and analyze the information in the column, and it's going to use it to build up this structure here that's going to include basically the matches to, so you have it's going to build up this matches of these different amino acids and their frequencies of occurrence in every position, or an insertion or a deletion. And it's going to encode that structure of that variation and the frequency of these transitions into this hidden Markov model. And so then you can take your candidate sequence and you can just thread it onto this Markov model, one nucleotide at a time, and it will take a look and it will say the odds of whether that sequence that you have provided to it is a member of the family. So it's a log odds store. So what's the odds that these statistics would have generated the sequence that you're looking at? And so that's essentially how it works. It's pretty accurate, except that you don't have strong signals for the start and stop for RNA operands. So when it's learning, it's basically doesn't really have a good idea of where to start or where to stop. This is in contrast for things like protein coding sequences, where the start signal, which is a start codon, and the stop signal are extremely strong signals, right? So you get sharp starts and stops, but for HMM predictions for RNA, for ribosomal RNA, it's a less, you have a bit of a spread, but the spread's not bad. And it works pretty good for bacteria. Okay. So functional annotation is this process of trying to attach a description to our protein coding sequences. So I mentioned, in contrast to the tRNA and the ribosomal RNA, just because you have a predicted an open reading frame that's predicted to be a protein coding sequence doesn't mean that it is well, you have it, but you don't know what it does, and so you're going to want to figure out what it does. Simplest way to do that is to just check against the database. So we're going to do a similarity search against the database. And if you find a good match, highly related, well-annotated reference sequence, then you can say, okay, well, it's close enough. I mean, if it's not 100%, then it may not be the same function, but if it's really close, and this is typically what's done, you say it's really, really strong match. I'm going to assume that this function is the same as the one from my reference database. I'm going to assign it the one from the reference database. So that's called transitive annotation. And if you can't find a highly related reference sequence to that matches, then you can go to some other kind of more voodoo approaches to generate like what are called observations. And a couple of observations might help you to ascribe a function, the general overall function to a protein coding sequence, but maybe not. The typical there are these processes or programs. One uses hidden Markov models. Is called PFAM, so is used to identify protein families which have certain functions, known functions. So you can use that if your blast against the database doesn't work. And these automated genome annotation programs will typically incorporate a number of these different um, programs that will consider their observations jointly to try and describe a overall function and use that as the annotation for your newly sequenced and annotated genome. Okay, so the sequence similarity is done by BLAST, and I don't think we have time to go through BLAST, but how it works. But what's important to notice is that it's very fast, which makes it very useful, and it can provide a score that gives you an estimate of the relatedness. That's called your expect value of the match. And so if the expect value is low enough, that's because the lower the expect value is, the more unique your sequence is, the less chance that you would have got a hit by a random hit to your database. So if you get a low sequence score, that implies that it's a unique high hit, and so that you can use the BLAST score as a measure of the reliability of the annotation that you're transitively assigning. Okay. And then you can use these BLAST databases that are helpfully generated for us by NCBI to help out with the annotation. So these are going to be the databases that we're going to consult to find out what the descriptions are and the possible annotations are from our newly generated and newly annotated genome. So the NR database is compiled by NCBI as a protein database for BLAST searches. You can download it and you can blast your candidate coding sequences against it and see if you get some hits. But it is not, and there's a nucleotide version called NT. That's the untranslated version. So this is a very broad database, but it is not expertly curated. So there's some question about to the accuracy of the annotations. But it's so you might want to use it, but if there's a better database, you'd want to use that first. So RefSeq database is a good example of a higher quality database. So there's less annotations, a smaller database, but the annotations in there are more reliable. You can typically, if you find a good match, you feel confident that the annotation that you're going to assign from the database to your CDS is correct. There's also this SwissProt or /UniProt database, and that is a very small database, but very high quality, manually annotated database. This is typically the first choice that is used to assign annotations to the candidate CDSs in your newly sequenced genome. And so it contains the functional description, but also a bunch of additional observations about your sequence. So this implies a strategy for annotation where you have your candidate gene and you want to extract all the high quality annotations and then progressively go to lower quality annotations, but where there are more of them. So the first thing you want to do is to look at UniProt, and if you can get a UniProt hit, and it's high quality, you can borrow its annotation. If not, then you can go to RefSeq. If it has a hit, borrow its annotation, right? If you get a hit, but it doesn't have an annotation associated, and that happens a lot, because these databases are constantly being supplied with newly sequenced genomes where you actually don't know the role, but you just know that it is in there and it has a match to something that's been seen something else, but nobody has managed to actually assign a function to it. You can just call it a conserved hypothetical sequence. You'll see that show up as a description a lot. So if it has an annotation, you borrow it. If it doesn't, but it has a hit, you can say it's a conserved hypothetical sequence. If it doesn't appear in RefSeq, then you can try NR. So there's more of a Hail Mary. It's a huge database, lots of descriptions. Maybe some might be wrong, but hopefully you've annotated maybe 50% or 60% of your candidate genes with your first two, and now you're just going to try to mop up the rest with NR. And so if it has an annotation, same process. If it has an annotation, borrow its annotation. If not, you can call it a conserved hypothetical sequence. And if you have a sequence that has no hits to any database, you can just call it a hypothetical sequence. So it was predicted to be a coding sequence, but you don't have any hits there. Okay, and that's it. I include for you, I'm not going to talk about these, but we're going to use them, reference them a little bit in the lab after this, is these main formats for recording annotations for genome. So one is the GenBank format. It has this header section, and this is the meta data for the entire record. And the more important one for our purposes is this feature table. Remember, features anything with a start and a stop. So, and something interesting that you can describe. So we have a feature table. There's different types of features. Genes and CDSs are common. So, but mRNA and tRNA would be other different types of features. So you have these feature keys that show up here. Each feature key is going to have these locations and qualifiers. So here is a feature, CDS feature, and it has a bunch of qualifiers, right? One is gene, right? Another is for here is like product, right? So you have a bunch of these different features. And then after your feature table, you'll get your sequence data down there. We also have this GFF3 format. This is a general feature format. This is just a general compact way to store feature data. So this is from this same GenBank file, just converted into this GenBank or just this GFF format, but it's basically a table format that has these 9 columns, and I list for you what what each field is for each column. Okay, but